NCRT diagrams, class 11 biology. So we have the structure of mitochondria, longitudinal structure. So the outermost layer is the layer which is outside is outermost membrane. Then we have inner membrane. Inner membrane forms the infoldings called cristae, which can be seen here. Then we have matrix. Matrix contains 70s ribosome, single circular DNA, and ribosomes are produced. Then RNA produces RNA is present, and ribosomes are also present in the matrix for protein synthesis. Then outer membrane is continuous limiting boundary of the organelle. Next is the sectional view of chloroplast. So we have the outer membrane, then inner membrane, then we have granum, then stroma is present, and thylakoids are the flattened membranous sacs. Then we have the stroma, in which dark reactions occurs. We have the stacks like piles of coins called grana. So this was all about the structure of chloroplast. We have region of root tip. So we have the root cap, then region of meristematic activity, then the long region is the region of elongation. Then we have root hairs. No, first we will have, in order we will have a region of maturation, then root hairs. Root hairs are formed from region of maturation. Root hairs are unicellular, and region of meristematic activity has dense protoplasm and it is vacuoleless. And we have region of elongation which have large vacuoles. Yes, and as we go upper it is called proximal, and as we go below it is called distal. Next is the structure of dicot seed. So we can see the dicot seed here. Outermost is the seed coat. Then in text it is given that micropyle is present above hilum, but look at the diagram, it is given below. So we have the cotyledon plumule on the upper side, radicle on the below. Next we have the structure of monocot seed. It is maize seed. So you can see the embryo, then endosperm. Then we have the fruit wall and the seed coat. Then endodermis. Then we have the alluron layer which is proteinaceous and 3 ploidy. Next, endodermis also has 3 ploidy. Then we can see the scutellum, and then coleptile, then plumule, then radicle and coleorhiza. Next diagram. First, this flower is the hypogynous. Then we have these two are perigynous, and then we have epigynous. This is marginal placentation, this is axile placentation, then it is parietal placentation, free central, and then basal placentation. Marginal placentation, the ovaries are present along the ventral suture, ridge-like arrangement in the ventral sutures. Then we have the axile placentation, which can be seen in tomato, lemon, and China rose. Then parietal placentation, ovaries are born either on the periphery or in the central. An example is mustard argemon. Then we have free central, in which ovaries are born in the central, which is false DP is the trick, which is free central, dance primrose. And then basal placentation, marigold and sunflower. Next we have photosynthesis chapter, in which it is given the C3 cycle. So starting with the carboxylation, so rubisco helps in carboxylation. A ribulose 1,5 bisphosphate, it helps in carboxylation. Atmospheric CO2 and water comes in, and then after carboxylation 3-phosphoglycerate is formed. Then reduction, in reduction to ATP, in reduction to ATP and to NADP plus are used. Then we have, after reduction, triose phosphate is formed, and then there is regeneration in which 1 ATP is used. Next, so remember, firstly after carboxylation 3 phosphoglycerate comes, then triose phosphate comes after reduction. Next we have H and slack pathway. So we have a mesophyll cell, then bundle sheath cell. In mesophyll cell we can see the plasma membrane, then outer is the cell wall, then it's a plasma membrane. When atmospheric CO2 in the form of H2CO3 and atmospheric CO2 in the form of H2HCO3 minus and the PEP, which is phosphoenol pyruvate, both combines, and then fixation both doesn't combine, fixation occurs, and then C4 acid, which is oxaloacetic acid, C4 acid, then transport occurs, then C4 acid gets transported to bundle sheath cell, then decarboxylation occurs and C3 acid is formed and CO2 is removed. Then fixation occurs by Kelvin cycle of CO2, and then C3 acid is transported to bundle sheath to mesophyll cells once again, and regeneration of PEP occurs. So important point here was that the oxaloacetic acid is formed in mesophyll cell, decarboxylation occurs in bundle sheath cell, then regeneration occurs in mesophyll cell. Next, photosynthesis diagram of ATP synthase and chemoosmosis. So we can see the thylakoid membrane is given here. H plus is concentrated in the lumen from the stroma. Between PS2 and cytochrome B6F, we can see the plastoquinone. How to remember it? Between PS2 and cytochrome B6F, we can see the plastoquinone. Then between cytochrome B6F and PS1, plastocyanin is seen, and oxidation of water occurs in the lumen. And then ATP synthase is the CF1 particle, and CF0 particle is present in the thylakoid membrane. In CF1 particle, ATP is formed from the ADP plus phosphate. As light enters the P700, NADP plus is reduced to NADPH, and in PS2, when light reaches that time, oxidation of water occurs, and stroma have low H plus ion and lumen has high H plus ion concentration. Through the ATP synthase, the H plus ion from stroma are transferred to the lumen. Important point here is that between PS2 and cytochrome B6F, plastoquinone is present, and between cytochrome B6F and PS1, plastocyanin is present. Next, Z scheme of light reaction. In Z scheme, PS1 and PS2 both works. As light reaches the light harvesting complex, that time electron gets excited, and then it is transferred to ETS. Then again electron light reaches the LHC, which is 700, which is PS1, and then electron acceptor, it reaches the electron acceptor, and in ETS ATP is formed from the ADP, and in PS1, after electron is accepted, at that time NADP plus is converted into NADPH. Next we can see the citric acid cycle of respiration chapter. So first is the pyruvate. Then pyruvate is 3 carbon compound. Then it gets converted into acetyl coenzyme A. Then so it enters the citric acid cycle. So starting the citric acid cycle, first oxaloacetic acid combines with the acetyl CoA. As oxaloacetic acid is 4 carbon, acetyl CoA is 2 carbon. Therefore 6 carbon citric acid is formed. Then citric acid gets converted into alpha ketoglutaric acid, 6 carbon to 5 carbon, and a carbon dioxide is released. So first step of decarboxylation is citric acid is converted to alpha ketoglutaric acid, and NAD plus is converted into NADH plus. Then alpha ketoglutaric acid is converted to succinic acid. This is also a step of decarboxylation in which 5 carbon compound gets converted into 4 carbon compound, and also NAD plus is converted into NADH plus, and GDP is converted to GTP. Next, succinic acid gets converted into malic acid. Succinic acid and malic acid both are 4 carbon compound, and in this step FAD plus is converted into FADH2. Next, malic acid is converted to oxaloacetic acid, in which 4 carbon compound is again converted to 4 carbon. So in this step NAD is converted to NADHH plus, and in this whole process there are two steps of decarboxylation. So first step is from citric acid to alpha ketoglutaric acid, and next is from alpha ketoglutaric acid to succinic acid, and 3 points where NAD is reduced to NADHH plus. So the first step where NADHH plus is reduced to NAD is from citric acid to alpha ketoglutaric acid. Second is from alpha ketoglutaric acid to succinic acid. Third is from malic acid to oxaloacetic acid. So the only step in which there is no conversion of NAD plus to NADH plus is from succinic acid to malic acid. And when succinyl CoA gets converted to succinic acid, at that time also when GTP is formed here. Next, ETS. So ETS occurs in the inner mitochondrial membrane. Complex 1 is NADH dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is cytochrome BC1. Complex 4 is cytochrome C oxidase. Complex 5 is ADP synthase.